Hello, this is my recording of Lecture 5. Unfortunately, due to technical problems, I was unable to um, record my presentation yesterday, and for this reason, there I'm in a conference with no one else, just speaking to myself, and the people in the virtual world were maybe watching this on YouTube or downloaded from our Blackboard site. I will be going over the concepts of the fifth part, which is on preparation technologies, a continuation from the previous lecture. So the first thing we want to mention is the oxidative method, or wet oxidation. Wet oxidation is the treatment with an oxidant at 140 to 200 degrees Celsius. So it has to be something with oxygen, right? So normally used oxidizers are peroxide H2O2, which is in liquid form, most at normal pressure and temperature, and ozone or air. Ozone is at normal temperature and pressure a gas. So this depends quite often if you want to use a liquid. If you're using liquid reaction, then you want to use hydrogen peroxide, and if you want to use a gas reaction, then ozone would be better. If if you don't need ozone, air can also be used. Remember that on top of my head, I believe 21% of our atmosphere is at ground at sea level uh, oxygen, so you can use just normal air. It's a really good oxidizer. As I breathe, I realize that. Especially lignin undergoes degradation and oxidation reactions. Remember, lignin is that hard, three-dimensional chicken wire that holds cells together, and it's a very complex system. It's an exothermic process, so it mildly combusts. Oh, here's Christian. Hello, Christian. You're going to see lecture 6 again. Um, the combination of dissolution by hydrolysis and degradation reactions. Hydrolysis reactions deliver organic acids such as acetic acid, which work as an autocatalyst. So it works autocatalytically. That's a mouthful. Meaning that in acid hydrolysis, normally you add acid, but in an oxidative method, quite often the cell contains some organic acids. So when you break the cell, the acids are free, and you have an your a self-contained an autocatalyst. Uh, Hemicellulose is degraded to a large extent to sugar monomers, which is quite nice. Remember, hemicellulose is a branching. The cons is cellulose is partially demolished. So if your total yield of sugars could be down if you're going for sugars, and also it can form harmful byproducts for the fermentation. Organic solvents, such as we also call organosolv, and ionic liquids. Lignocellulose in an admixture with an organic solvent and heated for dissolving will remove lignin, which is normally a co-product or a waste stream, you could say, a residue, and sometimes cellulose. So normally this is what you want. If you add an organic solvent, and remember, very common organic solvents are ethanol, methanol, acetone. We also use hexane, chloroform. These are all organic solvents. They make the solution of some particles uh, dissolute. So the solubility could be quite high for lignin for example, in a liquid. And if you add a co-solvent, or if you add an anti-solvent like ethanol, methanol, acetone, etc., the solubility were lowered. And as you lower the solubility of lignin, for example, it will precipitate or crystallize, so that it will be removed. So that's the aim, the removal of lignin. Normally this is done at a temperature between 8 and 200 on an industrial scale, depending on the solvent used. And the variables are time, temperature, pH, and of course, the solvent you are using. Yeah, it's gone dark here. I need to move more. The lights might come back on. The next is enzymatic digestion, which is often in combination with other methods to increase available surface area and to remove inhibitors. An enzyme can be done in conjunction with a hot steam fermentation, so cellulose turned into glucose, and then glucose into ethanol. Commonly used enzymes are amylase, which you know breaks down amylose, because if you remove the A and add an O, then you have what it breaks down. So an amylase enzyme breaks down amylose, and cellulase enzyme breaks down cellulose. So these are very commonly used enzymes. You must do this under mild conditions, and I've been asked a couple times, well, what are mild conditions? A mild condition is at a relatively low temperature, so around temperatures between, well, it has to be anything above 0, so around 4 degrees, which will slow down the process, but normally it's around 20 to 25 degrees. You can go up to around, depending on the enzyme, 60 degrees, because the higher the temperature, remember an enzyme is basically 2 amino acids, and it will break it apart if the temperature is too high. The bonds between the 2 amino acids will break. It will hydrolyze. Another condition you need to watch out for enzymes is the pH. This is be very careful, because the pH will also hydrolyze the bonds within the enzyme. So depending on the enzyme and how susceptible it is to hydrolyzation, you'll want to have a pH around 5 to 8. That's a good rule of thumb. It depends on the enzyme. An example of this is paper sludge. So here you have cellulose production at 28 degrees Celsius using a solid liquid extraction, a culture broth containing cellulase. You mix it. There's a separation of the solids. This can be used for ethanol, and the rest can be used to produce more bacteria. I now enjoy you to go to this video on enzymatic breakdown, and I'll go ahead and just record it from
YouTube. Innovation Spotlight. Sunliquid process converts straw to biofuel. Obtaining biofuels from the sugars of arable crops is standard practice. A new approach which holds out huge potential worldwide is the use of agricultural residues as an alternative feedstock. For example, bioethanol could be made from straw from Asia's rice paddies, or residues from sugarcane in South America, corn stover in the United States, or wheat straw in Europe. The biotech sunliquid process now allows Clariant to obtain difficult-to-extract sugar from non-food feedstocks such as cereal straw, corn stover, and bagasse. The sugar is then converted to ethanol. This approach overcomes the food versus fuel debate and offers an efficient way to reduce greenhouse gas emissions in comparison to fossil fuels. Whatever agricultural waste is available in the region can be used. In Europe, that is predominantly wheat straw, which is supplied in bales and cut into pieces. The process comprises 4 steps, starting with mechanical and thermal pretreatment. In a tank, the straw is treated with steam under pressure, which reduces it to fiber bundles. The second step is enzyme production. Clariant produces the enzymes as an integrated part of the process itself, minimizing procurement outlays and making the overall process more cost-efficient. Initially, a small quantity of the straw fiber is separated from the rest, fed into a small reactor, and provides the nutrient medium for the microorganisms that are then added. Under ideal process conditions, these microorganisms, which are tailored to the raw material employed, rapidly produce large quantities of enzymes. The rest of the straw fiber is liquefied in multiple reaction vessels by means of these enzymes. Since the enzymes are specially tailored to the raw material used and the conditions in the reactor, the subsequent process is far more efficient than with standard, non-specialized enzymes. The enzymes release all the sugars available in the straw and convert them to the respective monomers, to the C6 sugar, glucose, and the C5 sugars, xylose and arabinose. The enzymes act as biological scissors, cutting up the long sugar chains. The insoluble lignin is now removed and employed as fuel to provide the energy required throughout the process. The sunliquid process is energy self-sufficient, requiring no additional energy from fossil fuels. Next, the sugar solution produced in step 2 undergoes fermentation. All sugars are fermented simultaneously with the help of specially developed fermentation organisms. In other words, the organisms take up all the sugars at the same time and convert them to ethanol, which is initially dissolved in water. Using these specialized fermentation organisms, about 50% more ethanol is produced than with comparable processes. In step 4, ethanol purification, the ethanol is extracted from the water. This is normally achieved through distillation, a highly energy-intensive method. Clariant has developed an alternative, very energy-efficient process that employs specialized adsorbers. These are porous, sponge-like structures that act like a sieve. Water passes through the adsorbers while the ethanol is first bonded to their large surface area and then separated off. The resulting pure ethanol can now be processed to produce biofuel.
Okay, I'm going to cut them off. Also, I was just thinking, you may not even be able to have heard that, so I wanted to give you the opportunity of seeing the video, because you may not be, if you're watching this by video, then you can't click on this link. But I have a few more videos. I'm not going to show them because I'm not even sure you can hear the videos on this video. So if you want to go see these videos links, then you'll need to download the presentation on Blackboard and click on the links to see the videos. So I'll continue. Just a couple more sheets. All right, so now we have we can compare different types of methods. We now have looked at quite a number of preparation technologies and pre-treatment steps. So we have mechanical, the steam pretreatment, steam explosion, batch and flow, acid hydrolysis, alkaline hydrolysis, oxidative, wet ox, thermal plus acid, thermal plus alkaline, thermal plus oxidative, thermal plus alkaline and oxidative, ammonia treatment, and CO2 explosion. So most of these, or actually all of these, are good for increasing the accessible surface area, which is great because that's the very important part. If you want to decrystallize cellulose, for example, or to do anything, you need to increase the accessible surface area for a conversion step, which is in lectures 6 and 7. Lectures 6 and 7. Decrystallizing cellulose, that's mostly a mechanical, but also ammonia. Remember that cellulose crystals can be either crushed or they can be do alkaline. Solubility of hemicellulose you can use steam or acid. Cellulose and hemicellulose are have carbon-carbon bonds with sugars. They're polymers of sugars. So using acid, but also heat and steam is a good way of breaking these bonds. CO2 explosion is also good. The best way of, as I mentioned here, of breaking down lignin is using ammonia, but also any kind of thermal or alkaline reaction will solubilize lignin into the water, or whatever solvent you're using, which you then can later I'll show you that you can get. Oh wait, the organic solvents. Using organic solvents, you have a biomass. I'm probably not making sense. You have a biomass. You extract the lignin as a first pre-treatment step, and then using organic solvents, you can get the lignin out. So you solubilize lignin, and then you use organosolves to precipitate it. For furfural, HMFs is something you don't want. So the formation of furfurols in steam, acid, and thermoplus acid is not what you want. So an alteration of the lignin structure is quite good. Here you alter it, but you don't solubilize it. So you break down the lignin, but you don't get rid of it. So it's not ideal. Lastly, a comparison of these preparation steps. A steam explosion is normally batch, but also continuous at significantly higher temperatures than the rest, which means it costs a lot of money for the energy. But it has no chemical materials you needed. It's just the steam and water. It removes hemicellulose, but not the lignin. The particle size is average, so the large particle size, the better. If you can use something large, then you don't have to grind it, which is a step that costs, again, energy and money. Maximize cellulose to about 80-90% for digestion. Very low waste, no corrosive solution, or very little, to the inside of the tank. The complexity is low, and you can apply it to a factory scale, so a very large. If you remember on the kickoff for the project in Bergen, they have a steam explosion factory which they use for chicken feathers. Catalytic steam explosion is using a catalyst like a metal or like a metal or an enzyme. It's at lower temperatures, so I wouldn't use an enzyme at 200 degrees Celsius, but a metal. Consumption of chemicals, yes, chemical materials. So you are at significant cost. It does remove some cellulose, a little bit of lignin, and again a good size particle size for the feedstock. It maximizes cellulose at 80 to 85%, not bad. A little bit of waste because remember you're consuming some materials. There is slightly corrosive, and complexity is a little bit higher because you now are introducing a catalyst, and not as large a scale. A pilot scale is about, I would say about the size of a room rather than the size of a factory. Maybe a large room. You could use pretty large pilots, even a garage size. Acid hydrolysis, again, batch continuous, pretty lower temperatures than some, but still quite high. You consume acids, so that's an extra cost. Removes hemicellulose, which is good. Not so much the lignin. It produces a small particle size, not necessarily good. Maximum cellulose 80-85%, again quite nice. Has a lot of waste, which is very bad. You are consuming a lot of copious amounts of acid for the hydrolysis. It can be quite corrosive. Of all of them, has probably the highest corrosivity because you're using acids which can break down the insides of your tubes. You need to replace them in your factory. Complexity is middle, and pilot to demi factory. On a large factory scale, it's not being used. Thermohydrolysis is batch, as the name suggests, quite high temperatures. There's no chemicals involved. It does remove cellulose, but hardly any lignin, because you're only using heat, and lignin can actually withstand quite a lot of heat, unless you oxidize it. Particle size average, good cellulose digestion percentage, low waste, low corrosivity because you're not using anything except for heat, and a lab scale, so not very large amount of quantities. Ammonia fiber explosion, again batch, 2 to 10 minutes. Much lower temperatures needed. You're using a lot of ammonia, so there's a lot of chemicals required. No hemicellulose, but specifically very high lignin. So this is a technique you want to use to get rid of lignin. However, some of your feedstocks, some of your biomasses in your groups don't have lignin, so ignore that. Particle size is small. The maximum cellulose, 50 to 90%, that's actually quite low. The waste is quite high because you're using ammonia, which is particularly not only expensive but bad for the environment. It's corrosive, but not very corrosive. Ammonia, not as corrosive as acid. Complexity is middle and up to a pilot scale, so that's about the size of a room or a large garage or warehouse, but not a very large scale factory, not tens of thousands of tons. All right, that's it for lesson 5. Please ignore this lesson 6. This is lecture 5. I will save this recording and upload it to Blackboard and YouTube. If you have any questions, you can email me about that.